Structure of bacteria. The bacterial cell envelope is made up of three layers. The first is glycocalyx, which is a gel-like sticky layer made up of proteins and sugars. It doesn't allow the toxin and other pathogens, that is the large size molecules, to penetrate into the cell, as well as it acts like a cushion and protect the cell from injury. The second one is cell wall, followed by the plasma membrane. A difference in cell wall makes two kinds of bacteria. One is gram-positive and the other is gram-negative bacteria. When you see a gram-positive bacteria, peptidoglycan layers will be more and it forms a thick layer, whereas in the gram-negative bacteria, a lipid layer followed by a single layer of peptidoglycan and it is followed by plasma membrane. So during a staining procedure called gram staining, the thick-walled peptidoglycan in gram-positive bacteria is able to retain the purple color of the crystal violet and appears purple under microscope, whereas the thin wall of peptidoglycan in gram-negative bacteria is unable to retain the crystal violet, and it takes up a counter stain called safranin. Hence it appears red or pink under microscope. DNA in bacteria is of two kinds. One is genomic DNA, another is plasmid DNA, and their functions also varies. So genomic DNA has instructions for growth, metabolism, reproduction, protein synthesis, and it takes care of the metabolic activities of the cell, whereas the plasmid DNA has the ability to resist antibiotics, and it gives the antibiotic resistance ability to the bacteria, as well as it helps the bacteria to adapt to stressful environment. It plays a major role in genetic exchange through a process called conjugation, and it also carries genes for toxins in the case of pathogenic bacteria. There is a structure called mesosome, which are infoldings of cell membrane and are seen as special inclusions. They are in the form of either vesicles, tubules, or lamellae. Actually, mesosomes increase the area for enzymatic activity and it helps in respiration, secretion, and reproduction processes. So the only cell organelle which the bacteria has is the ribosome. It is 70S type. It has two subunits. One is 30S and another is 50S. You may wonder if you total 30 and 50 it should come as 80, but here we are mentioning it as 70. S is Swedberg unit. It doesn't indicate the mass, it indicates the sedimentation rate during centrifugation process, and hence here 70S is not the weight, it is the sedimentation rate. So several ribosomes may attach together to form a chain of polyribosomes, and this chain will produce proteins after receiving the instructions from the DNA. Apart from DNA and ribosomes, some other structures called inclusion bodies and gas vacuoles are present. Inclusion bodies are storage units and are not membrane bound, that is, they lack membrane. It includes phosphate granules, cyanophagine granules, glycogen granules, and it varies according to the type of bacteria. Gas vacuoles are present in purple, green, blue-green type of bacteria and helps in maintaining buoyancy. In the case of motile bacteria, that is the bacteria which can move, has a structure called flagella, which is a thin extension from cell wall, and it is the longest structure that extends from the cell surface to the outside. It will be minimum in number. There is another structure called pili, which are hair-like structures. First, it helps in conjugation, and it also helps in attachment of cell to some surface. You can notice 1 to 10 pili per cell. There are another minute structure which are shorter than pili are found, which are known as fimbriae. These are hair-like protein appendages, mainly helps in attachment. You can see 200 to 400 fimbriae per cell.